The renin-angiotensin-aldosterone system is one of the most significant hormonal mechanisms regarding the control of hemodynamic stability. This is attained by the critical regulation of blood volume, fluid volume, systemic vascular resistance, and sodium-potassium balance, overall impacting on cardiac output and arterial pressure. Involving the crucial homeostatic mechanisms of renin, angiotensin II, and aldosterone, RAAS aims to elevate bodily blood pressure in response to decreased renal blood pressure. Juxtaglomerular cells are located in the afferent arteriole of the kidney and are categorized as baroreceptors. Subsequently, the detection of an alteration or decrease in renal perfusion causes these cells to release renin into the blood. This proteolytic enzyme is primarily responsible for converting angiotensinogen, a protein released by the liver, into angiotensin I through proteolytic cleavage. Despite the physiological inactivity of angiotensin I, it is a vital component in the formation of angiotensin II. In order for this process of conversion to occur, the enzyme called angiotensin converting enzyme, or ACE, is utilized. Found in the vascular endothelium, particularly in the lungs, the ACE cleaves off two amino acids to finalize the formation of angiotensin II. Angiotensin II is a major bioactive peptide of the RAAS, undertaking four leading functions in which all aim to elevate blood pressure. These main functions include vasoconstriction, the initiation of the secretion of aldosterone, activation of the hypothalamus, and finally stimulating the release of the antidiuretic hormone. The vasoconstriction caused by angiotensin II takes place in systemic arterioles and increases systemic vascular resistance and arterial pressure. Angiotensin II is able to stimulate the release of the hormone aldosterone by acting on the adrenal cortex, or more specifically, the zona glomerulosa. Following its release, aldosterone travels back to the kidney and causes an increase in potassium excretion, sodium reabsorption, and fluid retention, overall increasing blood volume and elevating blood pressure. Upon traveling to the brain, angiotensin II binds to the hypothalamus, stimulating thirst throughout the body and therefore increased water intake. Finally, the effector hormone stimulates the release of the antidiuretic hormone ADH from the posterior pituitary gland located underneath the hypothalamus. The primary functions of ADH are to prevent the production of urine as well as increase water reabsorption in the kidney by inserting aquaporins at the collecting ducts of the nephron. All of these mechanisms collectively operate to increase blood volume and subsequently elevate blood pressure. RAAS provides a large amount of significance and relevance regarding human health. The molecules involved in the system are not only responsible for the control of blood pressure, but also relate directly to arterial hypertension, leading to the requirement of pharmacological interventions. Despite acting to manage blood volume on a long-term basis, the RAAS can alter blood volume chronically and can be activated improperly in various conditions, leading to the development of hypertension. There is clinical evidence suggesting that the system controls more than blood pressure, as a RAAS intervention has been seen to lower mortality and inflammatory marker levels in hypertensive patients as opposed to placebo patients. It is therefore apparent that the renin-angiotensin-aldosterone system is a vital regulator of homeostasis in the body, as it ensures to elevate blood pressure in response to decreased renal blood pressure.